Hi friends, my name's Steven with Leviathan Snakes, and for this week's ball python breeding starter guide video, we are going to be talking about allelic genes, incomplete dominant genes, dominant genes. Pretty much it's the other half of our genetics video. So we already did the recessive genes. Courtney rocked it. Definitely check it out over here if you haven't done so already. But if you enjoy the content, if you enjoy the channel, please consider hitting the like button, leaving a comment below during the title sequence, and subscribing. So let's get to it. So as we've said before, these videos are for people who are just now getting into the hobby and they're trying to do all of their initial research. And when you are talking about incomplete dominant genes, allelic pairs, specifically within the ball python hobby, there are some genetic terms that we need to know, and there's some hobby terms that we need to know in order to accurately describe all of the different things. So first we're going to start on the ball python hobby side. If you've been looking on Morph Market, if you've been doing any research on your own, you've probably noticed that there are genes called Inchi, GHI, Mojave, things like that. As well as you'll see genes that are Super Inchi, Super Mojave, Super GHI. And these right here are incomplete dominant genes. And an incomplete dominant gene is a gene that is defined by having two separate phenotypes depending on whether or not the animal carries one copy of the gene or two copies of the gene. So just like recessives, so this is where we're going to talk about a little bit of genetic terms. If we were talking about this not in the lens of the ball python hobby, but through biology and genetics, you wouldn't call a homozygous form of a gene a super. Instead, it would just be the homozygous form. So you can have a homozygous Inchi, which is the exact same thing as a super Inchi. You can have a heterozygous Inchi, which is the exact same thing as just a normal Inchi. Now, the reason why we don't use homozygous and heterozygous within incomplete dominance is that they, within the hobby, are almost exclusively, there's one exception on this, used to describe recessive genes. So a heterozygous clown means that they carry one copy of the clown gene, but they don't show it, and a homozygous clown is just called a clown, and it carries two copies. Why this is important to understand both on recessives and on the incomplete dominant side is that if you have a heterozygous form of an animal, whether it is a Inchi or a het clown, whenever they pass along their genes to an offspring, they have a 50% chance to pass along that gene or nothing, or a normal version of the gene, whatever else is on the allele, the locus point. So when you are thinking of a super, think of it the exact same way as a visual recessive. If you pair a super animal to anything, every single one of the offspring will be guaranteed to get one copy of the gene that they are a super for, that they are homozygous for. So if you have a super GHI and you pair it to a normal, every single baby will be a GHI. There's no guesswork, there's nothing like that. And this is really cool and really powerful for the incomplete dominant genes because you are able to take out some of the guesswork, take out some of the odds when you are trying to do your pairings, because you know if you have a super that every single one of those babies will get that gene. Now, when you are looking at incomplete dominant genes, some incomplete dominant genes are really easy to tell between their heterozygous form and their super form. So we'll talk about asphalt, for example. In asphalt, it on its own doesn't really look like a whole lot, like somebody who is brand new to ball pythons might think of it as a normal, but there are telltale signs of asphalt. You have strong flaming up the sides, you have brighter colors usually, but when you have a super asphalt, the homozygous form of asphalt, it is so wildly different. There is no guesswork at all. It's just oh yeah, that's a super asphalt. And it makes it really easy to ID. Now, you can also have animals that are incomplete dominant, but they don't have as strong of a difference between their incomplete dominant, their heterozygous form, and their homozygous form, banana being one. So a single gene banana, a heterozygous banana, has like very, very distinct colors. It's super easy to tell what a banana looks like. But if you have a super banana, really the big difference between super banana and a regular banana is just that the super banana is like a little bit more clean and lighter, so they don't develop the freckles later on. So depending on exactly what other genes are in there, it can be harder to ID a super banana versus, let's say, a banana lesser. Just things to consider when you are looking at incomplete dominance. Just because they are incomplete dominants and they have a visual phenotype doesn't mean that it's always going to be easy to identify them. And this gets so much more true the more genes that you pack in, because genes can interact with each other in ways that you wouldn't necessarily expect. So if we were looking at a Mojave ball python and we were looking at a GHI ball python, and you put them together and it looks wildly different. But if you had, let's say, cinnamon in there or pastel, those genes can kind of get covered up based off of the interaction between GHI and Mojave, and it can make IDing them a little bit harder than it would be otherwise. So just when you are trying to understand the incomplete dominant genes, really the best way to go about this, in my opinion, is do a bunch of research. Like, pick a morph. Find every single YouTube video that you can. Like, if you want to learn how to identify Inchy, just look for every single YouTube video on the morph Inchy and like look for the signs, as well as when you are making your pairings, try to have like more simple offspring. And this isn't so much for like strategy, but it's going to be way easier to identify Inchy if you only have Inchy, Mojave, and normals as like your possible combinations, or maybe Inchy Mojaves in there too. But if you are trying to identify Inchy in high intensity orange dream, Inchy, Mojave, fire, yellow belly, to a orange dream, asphalt, Inchy, you might not know, when you have so many other genes in the mix, whether or not they actually have Inchy, or if it's orange dream, or other genes that are kind of working in similar veins that do similar things. So trying to start like a little bit slower just to get a feel for identifying clutches is going to be really, really helpful. When you can see a clutch and you can see that one had cinnamon, that one has cinnamon hidden gene Wilma, that one has cinnamon hidden gene Wilma calico, it makes it really, really easy to identify because you're able to learn what every single step of the combo looks like. Moving on to dominant genes. Dominant genes are very similar to incomplete dominant, and with one copy of the gene, you can show a visual phenotype. So some examples of this would be pinstripe. Pinstripe is probably the best example, where you can have a beautiful pinstripe with one copy, but if you put two copies of pinstripe in an animal, the homozygous form, it does not show a alternate visual phenotype. It just looks like a regular pinstripe. And this doesn't mean that it's impossible to have a homozygous form of pinstripe or a homozygous form of any other dominant genes, just that you're not going to be able to tell simply from looking at them. You will have to breed them out in order to prove that they are homozygous. And when you breed them out to prove that they're homozygous, the way that you're able to tell is when every single one of those babies carries the gene that you think is the homozygous form, the super form. That is how you know that one of the parents also is, or that one of the parents is homozygous, because if you have a homozygous animal, they have to pass on that gene. Now, this is an area where it kind of like changes up a little bit. A lot of people have debated about different genes being incomplete dominant, different genes being dominant, and I don't think that necessarily that there is a 100% like every single situation this is a dominant gene because it doesn't show a visual phenotype. And one example is the leopard morph. So leopard, for the longest time, was generally viewed as a dominant gene, because even if you could get a super leopard, it didn't really look any different at all from a regular leopard. I would say that up until this last year, really like for sure this last year, when Captive Bred Specialties made their leopard, their sunset leopards and their sunset super leopards, were they like for sure like, oh yeah, that is a different visual phenotype for the super leopard sunset versus the leopard sunset. And I'll throw up a picture up here for them. But just because in one combo you can see a different visual phenotype doesn't mean you're always going to be able to see that combo. And it's been very, very common for people to think that, oh, I've paired a leopard to a leopard. I think that this one is a super leopard, and it didn't prove out to be super leopard because there isn't a big enough difference in the appearance normally for them to be able to tell whether or not it has a different visual phenotype and therefore would be incomplete dominant. Dominant genes are interesting because just because the heterozygous form of an animal is viable and it shows a phenotype like spider, and a super form of the animal is not viable and it will like die in the egg and it looks like an almost all white snake, but it doesn't ever thrive. It either dies in the egg or very shortly thereafter. People will say that spider is dominant, and I personally disagree with this. I say that the definition of a dominant gene is that one copy of the gene and two copies of the gene in the same animal doesn't show a different visual phenotype. But because if you have two copies of spider, it is a lethal combination, as a different visual phenotype, and they don't develop, versus one copy of the gene, that does not make spider a dominant gene. Instead, that makes it an incomplete dominant, because once you double up on the spider, it changes its phenotype and it makes it non-viable. Another thing that I want to talk about is visual dominance aside from genetic dominance. So, visual dominance is used when you are describing, in the ball python hobby, a animal that has a morph that is so strong that it can overwhelm the other morphs in it, so that way a pastel, for example, is a bright yellow snake. But if you put pastel and banana is so visually dominant that even though it's a genetically incomplete dominant gene, that the visual dominance of banana can overwhelm the pastel, so that way you don't get any of the yellows that you would normally associate with the pastel gene. So when you are thinking about visual dominance, this is usually used when people are planning out different combos for their breeding strategy. So I would say to be mindful of what combos, what genes that you're working with are going to be visually dominant and just overwhelming to other genes, because if you stack a bunch of genes into, let's say that you stack pastel, fire, banana, Mojave, a lace or something like this, it can get so hard to ID these genes that you won't actually know if they're in there or not. The same thing can go with champagne. Champagne's another visually dominant. Sunset is very visually dominant. And these genes have such a overwhelming effect on the visual appearance of the snake that this can be really, really cool because they can make really, really unique looking snakes, but it can also make it tricky to find what combos work well, so that way you can actually see all of the different genes that are in the animal instead of it just muddying them all up and you aren't sure if they're there or not. Now, moving on to allelic pairs. So allelic pairs are generally used when you're describing an incomplete dominant gene or a dominant gene and a separate gene. So the example I'm going to use on this is Mojave and butter. Mojave and butter both have completely different visual phenotypes. They look different, and it's very, very easy to see how they look different. So I'll show you like some of our hatchlings that have Mojave and have butter. They have other genes in them too, but that way you can kind of see like, here's what a Mojave looks like, here's what a butter looks like. When you put a Mojave and a butter together, they share the same locus point on a ball python's DNA, which that just means is that, you know how when you have two of the same gene that you can carry up to 2 copies of it, you get one from the mom, one from the dad. These genes share the same locus point. So if you would have a super Mojave or a super butter, those genes reside on the exact same place in the DNA. So if you have a Mojave and a butter, they would link together and they would form an allelic combination that has, usually for allelic combinations, they have wild differences in the visual phenotypes. So the most famous of all of these is the Bell complex. So Mojave and butter together looks like a blue-eyed Lucy, and this is Louis. He is our butter Mojave. He's actually a het ghost, but that doesn't actually matter for this. He's a butter Mojave, all white snake with blue eyes, and he came from an animal that had Mojave in them and butter in them, and it's just wild on how much allelic combinations have, how much power they have. The other really, really famous complex for allelic combinations is the yellow belly complex. Yellow belly is allelic with asphalt, gravel, specter, spark, so many different ones. And that these animals end up making the freeways or the highways, and they make some really, really cool, visually stunning animals that are just mind-blowing. So when you are thinking about an allelic combination, usually they have a very, very strong effect on the phenotype of the resulting offspring. In addition, whenever you breed an animal that has an allelic combination, just like a superform, just like a homozygous form, those animals will pass on one of the two genes that make up their allelic combination to every single baby. So we paired Louis to our Firefly Calico female this last year, and every single one of the babies carried either butter or Mojave in them, period. There were no normals, and this is really, really cool, really, really strong breeding resource if you don't want to hit normals in your offspring. So something to keep in mind when you are planning out whether or not you are going to hit for a specific combo, because let's say that you are trying to get a Wookiee Mojave GHI to a lesser Firefly Calico. You may not necessarily want to do that pairing, because if you hit butter if you hit lesser and Mojave in the same snake, all of the other genes that are inside of it, whether they have Wookiee or Fire or Pastel or Calico, you won't be able to tell. So this is really, really important to consider when you're planning out your breeding, is that if you hit an allelic combination that's so strong that it covers up the other genes, you won't be able to ID it, and you won't really be able to sell it for the full value unless you are able to breed it out and prove that this animal has these exact genes. So just something to keep in mind. I hope you guys liked the video. If you did, please like, comment, subscribe, and if you have a question, if you want to reach out to us directly, head over to Instagram. We are at Leviathan Snakes, and we'd love to talk to you. Anyways, we hope you enjoyed, and we will see you next time.